Hello, everyone, and welcome to today's webinar, Quality Controlling Your Flow Cytometry Data for Improved Experimental Outcomes. I'm Susie Valdez of Laboratory, and I will be your moderator for today's event. Today's educational web seminar is presented by Laboratory and brought to you by BD Biosciences. To learn more, visit them at bdbiosciences.com. Now, I want to encourage you to participate today by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd now like to welcome our speakers, Dr. Enrico Lugli, a principal investigator and the head of Laboratory of Translational Immunology in the Flow Cytometry Core, and Bob Balderas, a distinguished fellow and VP Biological Sciences and VP Market Development at BD Biosciences. Welcome, Enrico and Bob. We may now begin the presentation.
Thank you very much for the invitation. So hello, everyone, and first of all, thank you very much for attending today. It's always a pleasure to have an opportunity to address flow cytometry. You know, over the last year or so, we've hosted a number of domain experts to help us address, you know, the many issues and challenges that we all experience in building and working with building panels and working in flow cytometry. And that goes beyond just the panel. It goes on to determining where you get information about antigen density. It talks about, you know, preparing a sample, how we set up our instruments, all those questions that come in every single day about addressing compensation as well as spectral and mixing and autofluorescence. Then when we combine that with what we've seen over the last just few years about moving our flow data and analyzing it with the new data tools that we have, it gets more and more and more complicated. And the quality of the data always has to be maintained at the highest level. Well, today, what we'd like to do is to continue on this theme based on the many inputs and the questions that just still continue to come in, and they're shared from scientists like yourselves, you know, all over the world. We will focus today on QC of flow data and how we move that forward towards, you know, improving the quality of the interpreted data. It's my pleasure, though, today to introduce Dr. Enrico Lugli, a distinguished professor and certainly a domain expert, you know, in the field of flow cytometry. Enrico obtained his PhD in immunology from the University of Modena and Reggio, Emilia, where he focused his work on T-cell immune responses and homeostasis during the course of HIV infection. Now, it was at that point in his career that he got a great opportunity, and he moved to the Vaccine Research Center at the NIH, and where he continued to study the molecular mechanisms involved in the maintenance of long-term T-cell immunity. But he was able to do one other thing. He was with many different, you know, scientists, where the NIH is a domain area, you know, institution where flow is taught and where flow is at the top of the list of the tools that scientists use, and thus how we use it and whether it be the types of instruments that we have, how we look at data, and really how they look at the experimentation. It's really critical. Well, following that work that at the NIH and also a collaboration with Thomas Waldmann, he moved on and obtained an independent position as the principal investigator of the Laboratory of Translational Immunology and is head of the Humanitas Flow Cytometry Core. It is my pleasure to welcome Enrico. And Enrico, you can move forward with your presentation.
All right, thanks a lot, Bob. Thanks a lot for the invitation. It's a great pleasure to talk about stuff that is really at the center of our interest in the lab and in the flow core. So can you hear me well? that working okay? Can you hear me? Yeah?
Yes.
Okay. Okay, so today I want to show you, I want to discuss a bit of the things that we have learned over the years, and I think that it's important to know about them just because we're now advanced technology, data analysis, obviously with the work of many people involved. And I think it's becoming increasing. The complexity is just increasing over and over again. So I think it's important to discuss what are the major caveats that we encounter when we develop panels and analyze those data. So, Bob, it will, as we said, it will be nice to chat. So, if you guys have any questions, please interrupt me at any time. So, I look forward to do more of a chat than like a seminar. So, it's supposed to be an informal discussion on flow. So, what I want to go through today is to discuss some of the major aspects and the major caveats that we encountered when we developed the complex panels and when we first approached the analysis of high-dimensional flow cytometry data by using computational pipelines that I have to say were initially developed for analyzing cycle data or single-cell RNA sequencing data, then we have adapted to analyze the FACS data. So first of all, probably the basis of what is important here is how to design a panel correctly. So I'm not going through all the technicalities of panel development because other people have done this before me in this platform or other platforms. I know Yolanda Manke, a previous colleague of mine at the NIH, has done this, or Pratip Chattopadhyay. So, and they're great teachers in this regard. We will touch also the problem of sample preparation because in some cases it can lead to incorrect data collection. Obviously, the data quality is also very important, and we'll see how data pre-processing can change your final results following some computational analysis. This is something we have published a few years ago. And finally, also something that we tend to forget is obviously the biology. So the biology is very important, and we shouldn't think we just have numbers in a matrix, but we're talking about cells and expression of proteins. And so we have to look at our data always in light of the biology that we study. I think it's important to mention a paper that we have published in Nature Protocols a couple of years ago with Yolanda Brumann as first author, and this was done together with the lab of Burkhard Becker in Zurich. This paper, I suggest you to read it, and I'll be happy to welcome suggestions and critiques. And it's a list of all the procedures that you could apply to develop, apply, and computationally analyze high-dimensional flow cytometry data. It has all the protocols that might be useful to generate your high-dimensional stuff. So here, if we start from the basics, so if on the top left, you see, for example, a comparison of a couple of reagents that we have tested, and the suggestion obviously is to use always the best reagents. So sometimes this is not always possible. But just to give you an idea of how two different reagents, the same clone, but in two different colors, they perform completely different. So on the left, we have a CD2750. On the right, we have CD27PE. So you can see, obviously, the PE is much brighter. So when you develop a panel, then you should just you should just choose the best reagent if you have that possibility. And obviously, one other thing that we do, we test every single reagent that comes to the lab. So we do a full titration to determine the saturating concentration, and here the arrows tells you what is the best concentration at which you should use that reagent. But sometimes that can be the procedure you use to prepare your reagents can actually change the performance of the staining, because on the top right you see a difference in not in the positive signal in this case of an antibody, but actually of the background that is increasing very much when you use a FOXP3 fixation procedure. So that might be important for detecting intracellular antigen, but has a drawback of increasing the background very much, and in that case, you can lose sensitivity in that channel. So in that case, you don't have very much possibility unless you change antibody combination or you don't do the permeabilization procedure. So another important thing we check very carefully is, especially when we do experiments in different days and different batches, so obviously we want to avoid a batch effect between different days, is to not have compensation issues. So this is something which is that we control very well. Back in the days, we encountered this issue, even though we're following the 3 major rules of compensation developed by Mario Roederer. So Mario obviously is extremely instrumental in this regard. Even though we were following those 3 rules, we actually encountered this problem. So, for example, when we every time we used a BV711 antibody, so it didn't matter on the recognition of the antigen, it was just the fluorochrome that was interested by this problem. So, when we compensate, we can use compensation beats, and in 99.9% of the cases, it works great. But in this case, it's actually it was always associated with overcompensation, at least in this combination of BV711 into the APC R700 channel. We don't really know the reason why, but we realize that if we use cells to make our single stain compensation controls instead of the beads, then the compensation was perfect. And on the right, you can see that both both single stain compensation controls were bright enough to allow you to calculate compensation. But it was just the binding of the beads that was somehow changing the interference and the contribution into the secondary channel. So now we overcome that by using cells instead of beads, where we need to compensate our panels, and then it works perfectly, and we don't have to worry about that anymore. So, but remember that you might have those issues. So now now that we talked a bit about the panel development, but again, I'm not going to go into all the details of the panel development. Now you have to get your data, you acquire your data, and you have to process your data for computational analysis. So here is always from our Nature protocol paper, an example of the removal of the unwanted events that we want to have before performing computational analysis. So you can see from the slides that in some cases we can have some antibody aggregates. So, for example, in the second row in the central plot, we can have just expression of some spurious events that are very bright. Those events are not really they should be excluded from the analysis, and in that case, sorry, having some issues with control of the slides. So the folks you can keep control of the slides if you want. It's fine, and then I will just say next, and you'll move to the next one. Okay, so sorry for interrupting. So we apply a very stringent gating procedure just to select the population of interest, and in this case, we can select just the population that is of interest for us. That is dependent on the biological question. So if you're interested just in the CD8, and that's something we'll see later in the presentation, then you can just select the CD8, or otherwise, if you want to have the full spectrum of immunity, for example, in a tumor, we generally include a CD45 antibody, and then we isolate all CD45 positive cells and computationally analyze those. But it's important to remove all the cells that we're not interested in and all the cells that can be a source of artifact, because if you have spurious events, for example, due to antibody aggregates or those, they don't generally they generally look undercompensated, but you shouldn't worry about the compensation of those because they just follow their own rules, and so you should remove those and not have them in your final analysis. And you can export all those files and then feed them into your favorite R script, and we're going to talk about that in a second. So how when we developed this Nature Protocols paper with Burkhard Becker, we also wanted to include a part on how to QC your data before feeding them into your computational analysis. So that's important because in the past we used to go through every single file, every single sample, for example, by using our favorite flow cytometry analysis software. So now we tend not to do that anymore because otherwise it would be like a huge waste of time. So obviously we check that everything is fine in terms of quality of the data, but we don't really check for the position of every single population. That should be done by doing a strong and very defined QC on your machine or on your panel development. So if you go to the next slide, slide number 7. So in slide number 7 here, you see that in this case we ran 3 different samples, one from a healthy donor, one from a cancer patient from the peripheral blood, and from the tumor of the patient. And we overlaid all the antigen expression. And so in most of the cases, you don't have like strange patterns. But I just want to highlight a couple of examples, one that is correct and one that isn't correct. It is incorrect, and you should ring a bell when you see strange things like that. So, for example, in the case of CD4, you can see that the peak of CD4 expression is exactly in the same place. It doesn't matter where it comes from. If it is from the healthy donor, the patient's blood, or the patient tumor. So this is important because from the biology we know that CD4 expression on the cell surface tends to be very stable. You have a fixed number of receptors on the cell surface. So when you stain with antibodies at saturating concentration, then you should see fluorescence always in the same place. So obviously you see a different abundance of CD4 expression because from the tumor we don't really have that many CD4s infiltrating the tumor, at least in this case. Instead, from the peripheral blood or the healthy donor, we have many more CD4s. But different is, for example, on the bottom left, this protein called GNLY or granulysin. So granulysin is a cytotoxic marker, and we know from biology that just a fraction of the cells express granulysin. So you should have the majority of the cells that are negative, and a bit of cells that are positive, and in general, the expression of the protein is a smear of fluorescence. So if you see the sample in red here, the patient's blood, it looks like the peak is all moved to the right. So ideally, you could think that all cells are positive. But we know that this is not possible because granulysin, if you know a little bit about T-cell differentiation, is expressed by terminally differentiated cytotoxic cells. They could be CD8 cells or CD4 cells. And so it's not possible unless it is a very strange sample that your patient's blood has a lot of granulysin being expressed, because there will be some naive cells, for example, which are at the very beginning of differentiation. And in that case, naive cells, by biology, they don't express granulysin. So it's not possible that we have a signal like that. So this should ring a bell. And in that case, we have a strange expression of a strange pattern of fluorescence for that antigen for that specific sample. So what do we do in that case? So in that case, before feeding all the data into the computational pipeline, one thing we can do is we can just remove that parameter from analysis. But obviously, so this will compromise the whole analysis for granulysin. Otherwise, if you want to keep this parameter in the final analysis, then you just remove that specific sample. So in this case, you need to find a compromise, because you can have it all unless you control for everything, but in some cases it's very difficult. So we don't know what happened in this assay, but we're happy that it happened. So we could give you an example. It was probably sample dependent, because one thing we did afterwards, we had a replicate vial from that individual. We followed the cells and we stained again, and we didn't see that problem anymore. So there might have been something that went wrong with staining for that specific patient's blood, and we don't really know the cause. It would be great to know the cause, but sometimes you just don't know. So you either repeat your experiment or you remove the sample from the analysis. So another thing that we can do is we can analyze the antigen expression. This is more intuitive, and it's also dependent on the experience of the researcher. But if you look on the right, you see a lot of t-SNE plots, and we overlay the expression of every single antigen, and we just looked at the expression of these antigens was co-regulated or anti-regulated, depending on the biology. So, for example, you know the CD39 is almost never co-expressed with 127. If you know the biology of T cells, so this is just a way to show you if the data follow what you expect. But obviously, don't always think that you will obtain what you expect, because otherwise, you will never discover anything. So just keep your eyes open also for the data that looks strange, because I think it's always nice to follow what is undefined. If we can go to the next slide. So thank you. Here, this is something that we encountered a while ago when we decided to analyze samples by t-SNE. This was one of the first times that we used t-SNE to analyze flow cytometry data. In this case, we did an interesting experiment, because we wanted to test the possibility to replicate data in different experiments over time. So here we took 3 different healthy donors that we run over PBMCs, that we run over 5 different experiments. It was a 27 parameter panel. We run over 5 different experiments in 5 different days. And we isolated the CD8 cells in this case, and then we run t-SNE by using the parameters that are indicated here. And then we debarcoded. So every sample had barcode, so after analysis we could debarcode and look at the t-SNE distribution over the 5 different experiments. And what you can see here in these 5 different experiments, we just draw a gate. That gate doesn't really identify anything, it just identifies a region. But you can see the percentage of the cells inside different gates was actually very different. So we thought, what is going on here? We might have done something very strange, or there is no reproducibility over the different experiments. If we can go to the next slide, please. So we thought about different possible sources of error. So you might have technical reasons, or you might have computational reasons of why we obtained that non-reproducibility. So first of all, we thought about instrument performance. That was unlikely, because we check instruments with rainbow beads every day. We imported the protocol that Steve Perfetto developed at the NIH years ago to track your instrument performance from day to day, and we were pleased to see, I don't have the data here, but it's published, we were pleased to see actually the performance of the instrument over these 5 experiments spanning a few weeks was actually rock steady. So it didn't really move. The instrument was very stable. So it wasn't, the instrument itself was not a source of error. We thought about the batch effect, like antibody staining or pipette, but we didn't really see any effect, because we didn't really see changes in the expression of the different proteins. Also, the staining was correct. Obviously, compensation. We have talked about compensation before. Compensation and spreading error that we knew is very important when we developed panels was similar across the 5 different experiments. But we realized that background fluorescence was different from the different experiments for some of the antigens. Okay, so we observed difference between samples, and we realized that if we by exponentially transform or logically transform the samples before feeding them into computational analysis, then we obtained, as you can see in this TISNE here, very good reproducibility from one experiment to the other. So you can see that the plots look very much similar from one to the other. So sometimes you see some movement clouds, but this is due to micro differences in the MFI from experiment to experiment that maybe you want, if you want, you can consider to normalize if you have always the same sample, the same reference samples run across the different experiments that you're analyzing. So these are some of the difficulties that we encountered when we first developed complex panels, and we wanted to analyze them by computational analysis. So I just want to move, so if there is any questions coming up, I'll be happy to answer. Otherwise, I'll just go on and discuss all the display of these high-dimensional data that we obtained by computational analysis.
Yeah, Enrico, we'll save these questions for a little bit later. There's a number of them.
Okay, so I'll just keep going. So if we can go to the next slide, I'm going to introduce different ways we use to display high-dimensional single-cell data. So if you can go to the next slide, this is a study that we have done a few years ago. Again, Yolanda Brumann is first author, a great postdoc that stayed in my lab for a few years. And when first of all she pioneered the optimization of this technology in the lab, and then used it to analyze the infiltrate of non-small cell lung cancer samples. This was a collaboration with a thoracic surgery in our institute. So she took 53 different tumors and paired lung tissues, and at the same time she analyzed 23 different bloods. She processed the single-cell suspension, and then she analyzed the CD8 cells. Here, as I said, we didn't do standard gating anymore. So we didn't gate on the first 2 antigens, and then defining a population, and then going down with 2 more antigens and 2 more antigens. Here, we decided just to take all CD8 cells from every single individual. Just for speed, we took 3,000 cells per sample. We probably didn't need that many cells to analyze, just to have an idea of how the data look like. We isolated all CD8 cells. We barcoded them with a unique barcode, so we could go back and look at every individual sample. And we put them together into a single huge file. And this was a big file of many hundreds of thousands of cells that we analyzed by clustering algorithms. And these clustering algorithms were able to give us a number of different clusters that we'll see in a second. And these clusters basically have a certain phenotype that you can determine, and then you can use your favorite data display method. And I'm going to show you 2 or 3 of them just to have an idea of how we show the data. So if we can go to the next slide. This is just a heat map. I'm going to walk you through this. The heat map shows you the antigen expression in the different clusters. You've got the markers in the columns, so you can see CXCR5, CXCR3, CD73, and so on. And then you've got the phenograph clusters. So we use this computational algorithm called phenograph, developed initially for single-cell RNA sequencing. And you can tune the parameters to obtain less or more clusters depending on what is your idea of the data. But in this case, we had more than 20 clusters that we obtained, and there is just to have a little bit of granularity on the data. So now then we meta-cluster both the markers and the clusters. So all the different markers and the different clusters are put together according to their similarities and differences. And the colors indicate the abundance of antigen expression. So in this case, we used an approach that we imported again from Mario's lab called integrated MFI, which is just the MFI multiplied by the frequency of positive cells. This can have can have advantages and disadvantages as any display method. So any display method might have limits. But I think it's intuitive on one side, and it's also indicative of the protein abundance per cluster. Okay. So, for example, imagine having all these clusters. You are able to sort them, and you do RNA sequencing on all of them, and you will have somehow a similar result. So, the abundance of protein expression in this case, or RNA in the case of RNA sequencing, per population, and this is more or less the same thing we're doing here. So, you have an idea of the abundance of protein expression in every single cluster you identify. So there might be a disadvantage. So the calculation may be biased by the frequency of positive cells or the MFI. So for example, if you have few cells with high protein expression or many cells with low protein expression, you might end up with the same result. So in the end, this is great to walk you through the data to give you an idea of the trend that you have in this big data. But at the same time, it's always great to go back to the raw data and use your standard methods then to analyze the data and confirm. This is a very good way to have a fast idea of what is going on in your huge data set. As I said in the very beginning, so we barcoded every single individual, so we could then debarcode in the analysis. In this case, we just debarcoded in the 3 different tissues. In this case, it was the blood, the normal tissue, and the tumor. And then by using these balloon plots, we displayed the abundance of every single cluster, and we could do our favorite statistics to see what was the difference in the abundance between the tumor and the blood or the normal tissue. So if we can go to the next slide. This is just a different way to show data. So it's a different panel, but more or less it gives you similar results. So instead of using a heat map, here we used a balloon plot where the balloons are the size of the balloon is indicative of the percentage of positive cells, and the color is indicative of the MFI. So the bigger the balloon, the more cells. So this solves the problem that we had before. The bigger the balloons, the more cells express that antigen, and the more cells, express that antigen, and the darker the color, then the higher the MFI for that specific antigen in the different clusters. Again, you can meta-cluster these clusters and the antigen as well, and by using your knowledge, now you can name all the clusters that you obtain. On the right, we have just bar charts depicting the frequency of every single cluster among the different tissues. Also, in this case, we had a blood tissue, normal tissue, and the tumor tissue. This was from a breast cancer, a paper that Agnese Rosurdo published in my lab. What is the advantage of this one, of this display from the right, is that you have an idea of what is the frequency of a given cluster across the different tissues. But at the same time has a disadvantage because it doesn't give you a definitive answer on the abundance across all the different clusters. So you don't know if that cluster is actually rare or very abundant. So also, again, it can solve some problems, but at the same time, you might have limitations on other sides. So it's always important to consider all the different display methods and be aware of the limitations that you might encounter. If we can go to the next slide. This is the same data from the lung cancer study. In this case, we wanted to depict the cluster abundance that we obtained by phenograph clustering in all the individuals that we analyzed. So showing individual samples can be very important. It's important to see heterogeneity. It is important to see what is the heterogeneity in your cohort of individuals, and at the same time to see how the different tissues look like, if they're similar or not. So in this case, for example, We did meta-clustering of all these clusters, and we can see that the blood samples in blue, they all cluster into a branch of the tree, and the tumor in a different branch of the tree. Instead, the normal tissues are scattered between the blood and the tumor tissues. So some tissues look like more like the tumor, and others more like the blood. The other interesting thing, you may identify subgroups of individuals. So in this case, you might think maybe there might be heterogeneity, and maybe some patients are different from others by looking at the immunophenotype. In this case, it was the CD8 T cell phenotype as a whole. The other important thing, by using this method, you don't have aggregate data, but you have data from every single individual. You can see if there is a specific contribution from one single individual to the identification of a given population. So, for example, the cluster that we see in orange, you just have it in a very specific set of blood samples, but not in all the samples. So that means that the contribution of that cluster will be from 2 or 3 individuals, and not from the entire cohort. I think that's very important, because otherwise you might be driven by the wrong data. When you have cumulative data or summary data, in this case, you can clearly see what is the contribution of every single individual. So finally, if we can go to the next slide, I just want to talk about the power of integrating single-site technologies. So that's something that we have discovered in a recent Nature Immunology paper that we published last year from Giovanni Galletti and others. If we can go to the next slide, please. So here we have done, we started in an unbiased way. So the flow cytometry is great on one side, because you can measure, at least by using the technology, we use up to 30 parameters on single cells, and which is not huge, which is not small, but at the same time it's not huge. But I think it can be very enlightening. And at the same time, you can measure millions of cells, so that's the very good thing. Nevertheless, there is now technologies that can measure thousands and thousands of genes from a single cell, such as single-cell RNA sequencing. Many people use it now. So at the time, we thought, okay, so let's have a look at the peripheral blood CD8 positive T cell compartment. If by using this technology we discover something new, which is not always the same subset. So here you have a different clusterization of different cell subsets. We were interested in cells of the early differentiated compartment, which are the clusters at the top. I'm sorry, the animation is broken here, but the clusters at the top identify early differentiated memory cells. But the important message here is that we identified a protein called granzyme K that was differentially expressed between these two clusters. And we used that to design a panel along a number of other antigen receptors to develop a high-dimensional flow cytometry panel. If we can go to the next slide. So here, by using granzyme K, we were actually able to distinguish CCR7 positive, granzyme K positive, and granzyme K negative cells. The interesting thing is by including antigens that we thought were probably important for T cell differentiation, we were able to show that these granzyme K positive cells, which at the first glance look like effector cells, actually express inhibitory receptors such as TIGIT and PD-1. And that's something that we didn't see by single-cell sequencing. And the reason why, I don't know. I guess it's because the expression in the peripheral blood at the level of the mRNA is low, or the other possibility is that the technology is not powerful enough to see those mRNAs. But when we used flow to detect protein, that we saw a very good protein expression. You can see here in this UMAP. If we can go to the next slide, it's just the raw data to show you that we can have very bright PD-1 and TIGIT expression in the peripheral blood. And on the left, you have the naive control, which by biology shouldn't express these inhibitory receptors. We found that the stem-like memory cells, a population of cells that I discovered together with Luca Gattinoni when I was a postdoc in Mario's lab, expressed some, and central memory cells were actually the ones expressing the most. So this was the foundation of a new study that led to the identification of precursors of exhausted cells, which is something that we have never seen before in the peripheral blood of healthy individuals, and we studied them at the epigenetic and functional level. So just to wrap up, if we can go to the next slide, please. I just want to give you the most important take-home messages from this webinar, and I'm happy to take questions afterwards. So computational analysis are great tools that we can use to rapidly investigate trends in the data. Okay, so we use gating now to confirm the data according to established definitions. So, for example, if we identify cells that look like Tregs, then we can baptize that cluster as a Treg, but then we have to go back to the raw data to say that CD25 positive, FOXP3 positive are actually increased or decreased in a given cohort of individuals. The advantage of these tools is huge. The effort and time is very different. We can it takes hours to analyze huge amount of data. So, in generally, it takes let's say a few days to develop a panel, a few days to run the samples, a few hours to analyze the data, and probably a few weeks to interpret the data. I remember when I was a postdoc and I used to do gating in dozens of samples, which is not a huge number nowadays, and it took me weeks to go through the whole study, and now within hours we can get an idea. We can have an idea of how the data look like. But remember, there is many possible sources of error, probably more than those I have mentioned here. In computational analysis, the machine performance is very important. We QC instruments every day and before every experiment we do. So that's something that I will thank forever Steve Professor and Mario they taught me how to do it. So panel development is a possible source of error. So we have published a paper in that sense, but there is many more papers that you can follow in that regard. The data pre-processing, we have seen that it can be a source of error, and obviously it's sample handling, which can generate artifacts. My suggestion is, so our Nature Protocols paper contains instructions on how to use phenograph or FlowSum, another clustering algorithm developed by Ivan Seitz in Ghent. But you can use many different pipelines. There's actually now a new pipeline every week. So most of these are developed for single-cell sequencing or cytokine. They must be optimized to handle fax data. It might take a while, but I guess it's worth the effort. And if you want to explore more what we have done so far, you can get our scripts. They're all available in GitHub on the Lugli Lab page. So also, if you want to reach me by email or on Twitter at Lugli Lab, you can chat with me, and I'll be happy to provide you with suggestions or expertise if you need any help. So next slide, I just want to thank all the people involved in this. I mentioned many times Yolanda Brumann, who pioneered the optimization of these technologies first in our lab, and Georgia Lisi, who has been supporting her very much, and the computational biologists in my lab, Emilia and Simone Puccio, have done a lot of work in this sense, and all the collaborators and the funding. So thank you very much. Thanks, Bob, and I'm happy to chat more about this.
Thanks, Enrico. Thanks very much. I think, some of your comments, at the end that you brought in about the experience that you had and got from Mario's lab about making sure that instrument is like QC'd every day is always a reminder to everyone. But, I think we all know that in so many labs around the world and core facilities, there are wonderful, domain individuals that take care of these experiments for us all the time and maintain those instruments at the highest level. And when they are, then we have a greater chance of really great success. But Enrico, we have a number of questions, and I think for the questions that are coming in, just to let everybody know, if we don't get to them, we will address them, and we will get in contact with you and make sure that the specifics to every question are addressed. But we have maybe Enrico, we have a number of questions, and I think, we can take them from many different directions. But, as we go higher and higher in plexing, with the types of technologies that we have today, and we're faced with situations like fluorescence -1, and
right.
When it's doable, right? When you got 10, 15 colors, right, to work with. And what do you see? What should we do as we approach 25 and 30 and 35 colors, and maybe don't have enough cells to really evaluate that sample from every direction?
Right. So first of all, the fluorescence -1 controls, they're great to define where the positivity for antigen is. Okay. So the one thing we have to keep in mind is also they tell you if the compensation is correct. So if you have problems with compensation, fluorescence -1 control will tell you if it's working or not. And obviously, the other thing is if you have a lot of spreading into a given channel, that might be important. So we tend to do that for antigens that we don't really know the pattern about. Okay, so if you have, let's say, HLA-DR expression, HLA-DR is in most of the cases a smear of expression. So in that case, we tend to do FMO controls. But if we have antigens that we know very well and are generally separate very well, we don't do FMO controls in that case. The other important thing I want to mention, because we tend to forget about this, so you must be carefully using. So FMO is the control where you have all the antigens except one. Okay, except one antibody. Your antibody that you're removing in the FMO control must be titrated carefully, because if you add that antibody and then your background goes higher, just because it's the nature of the antibody giving more background, well, in that case, the FMO will be used. So I think that's important to remember.
It's just a reminder of how you balance those experiments, and I appreciate that side. One of the other things that we get asked about, right, all the time is as we increase the number of let's say colors in an experiment, we're adding more and more and more antibodies to it, and we get to that point where we're at the mercy in some ways of the concentration of those reagents when they're bought as single vial reagents. And they have in the human side, right, we still work to a certain percentage of them still in test size and not in mass size. I guess maybe just a question for you. Do you think that all reagents in flow should be converted over to mass units like we do in the mouse field, especially for these high dimensional experiments?
Do you mean micrograms or
Yeah, in a microgram format rather than in a format of test size, or do you not see the problem associated with keeping saturation at a specific level?
Well, no, saturation is important. We always titrate just to make sure we use saturating concentration, because so in some cases we have a lot of samples to stain, and we don't really count the cells, so we don't really know how many cells we're staining. So we just have to make sure we are in a good range of concentration, so we saturate all the receptors. Ideally, to have the mass, I don't know. We don't really look. So to be honest, I don't really look at the data sheet when I get a reagent. I just go straight to titration and then test, and then see what is the optimal concentration. So in the end, we test everything and make sure that everything works correctly. And if we change antibody, we change lot. We just have to make sure it performs the same way as the one before. Okay. So that's very important for reproducibility.
Yeah, I think I appreciate that. One of the big questions that also comes out all the time is, with all the data tools that are out there, how do we evaluate the accuracy of high dimensional flow data?
So I tried to explain a little bit during the webinar. So there's many QC steps. Okay, so it's not a simple work. I think so for example, as we have an RNA-seq, and then the bioinformaticians go through a number of different QC steps. Flow cytometry somehow doesn't. Things are changing, but it doesn't really have this level of let's say control, because ideally the technology is simple. You get some cells, you transfer some antibodies in, and then you run through the flow cytometer, which is easy to use. It's not sequencer. That is probably more difficult to use, so you have specialized technicians doing that. But from let's start from the basics. So antibody titration, make sure your antibody is validated correctly. And then you put together some antigens, and you have to make sure that they don't interfere too much with each other. Or if they do, then you have to make sure you can manage that interference by designing your panel correctly, by making sure you can cope with the spreading error. And that's the second level. And then you run a complex panel, and maybe you do comparison with a less complex panel just to make sure it performs the same. And then once you run all your experiments, you have to make sure you run today, you run tomorrow, you have similar performance. So machine performance, also sustaining performance. And once you have done all that, then it's already quite a lot. Okay, so sample preparation and panel development is very important. And then with the high dimensional data, then it's also a matter of optimization in that case. So in the very beginning, we were running these KNN clustering algorithms, and so we had to go through all the different K just to decide what is the correct number of the nearest neighbors. I don't think there is a definitive answer. So now we tend to run to analyze the elbow plot to show you what is the optimal number of cluster per different Ks. But in the very beginning, we used to run like at K, let's say 15, 30, 100, 500, and then analyze all the different heat maps. It was like a lot of work just to make sure that we're making sense. Okay, and then going back to the original data and confirm. So this is explained quite a bit in our Nature protocols. It's quite detailed in that sense. But now it's been 2 years already, so there are a lot of new developments. So if you can find maybe tutorials from other computational algorithms, they're probably even more advanced.
Oh, very good. One of the things that I'm sure you saw and were really involved with at NIH involved a lot of determining the minimum number of cells, so like the antigen-specific responses you did to the HIV peptides and all that work that was done, and we saw how many cells that we had to obtain just to get the accuracy of data. But today, we're using so many different antibodies and evaluating so many different dimensions of biology. How do we evaluate the least number of events you need to get quality data?
Okay, so again, there is a very nice comment from Mario in Cytometry about probably more than 10 years ago. It's called "How many cells are enough? Are you positive?" or something like that. I think it poses the problem. So obviously, it depends on the frequency of the cells that you're looking for. So if the cell is antigen specific, you might want to use a lot of cells, because otherwise you won't be able to detect that specific response. Otherwise, if the cell population is very abundant, then you don't have to run that many cells. We can easily do phenotyping on few hundred cells from tumors. So obviously, the robustness of all the clusterings, if you want to obtain a lot of clusters, then it's not very robust. But if you're looking for the major populations, a few hundred cells is fine if the distribution of your antigen is abundant enough. So it's like vaccinating a large population of people, right? So you vaccinate 1,000 and you don't get side effects, and then you vaccinate 1 million and you get one side effect in 1 million, which is still very low. Just to bring it to today's news, so get vaccinated or run a lot of cells if you have the possibility.
Yeah, it's a real problem with a lot of these experiments that we have today. You know, there's other questions in here, and there's just many from so many directions. But you know, when you sit down and you begin to process data and to look with these data tools that we have today, you know, how do you go through the selection process of determining which one of the algorithms that you use to analyze data?
So this is something that some people do, but not many people do. So again, there is like a new algorithm coming out every week. Okay, it's really overwhelming. It's a lot of stuff, which is good because then you can test many of them. But at the same time, you need to get your answers in the end. So I don't think it's feasible to test a lot of things. So we started working with this algorithm called Phenograph a while ago, and we like it, so we kept using it. But for example, I also mentioned FlowSum from Ivan. So Ivan developed this very nice algorithm, which is also faster than Phenograph. But there's others. You know, they're not the only ones. We try to combine accuracy with speed. Also, some might be very fast but not very accurate, or they might be very accurate but not that fast. And then if it takes days to get the data back, then I don't think it's very feasible. So you do this because you want to speed up discovery, and so you have to find a compromise. In general, you know, even though it might take a few hours in some cases, but now we have, for example, Simone developed a Python code that now takes minutes instead of hours. So it's much faster. But otherwise, we run them like at night, so the computer just goes on and we sleep. So
that's good. You know, we just have a minute or so left here, and I know it's certainly something that many people are looking at today, and ourselves internally. Even when we QC and look at panels and try to understand them, as we look at biology and we expand the numbers of specificities and we define biology in so many different ways today, we're finding that the repertoire of normal biology is just really, really, you know, complex, and there's receptors missing, and not all people, as we know, are not the same. But what do we need to do as we address a disease, and we collectively look at lots of different, you know, samples, but are we looking at enough normals to understand really the receptor proteome in normals before we go through and just look at a lot of diseases where we too see these types of deletions of certain specificities? Are we doing enough in normal biology?
I think we lost him.
I think we lost Enrico on that last question. I actually really wanted to hear that answer. I guess in closing, you know, I'd like to say to everyone that we appreciate very much. We'll too try to get Enrico's answer on that last question, because I think it's a really important one for our field. And I'll turn it over to our moderators to close our session.
And I too want to thank Enrico and Bob for their time today and for their important presentation. And I also want to thank LabRoots and our sponsor BD Biosciences for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and stay safe. Bye-bye.
Thank you. Bye-bye.
Thanks, Enrico.
Yeah, sorry.